I can't hear you.
Your mic is muted.
Thank you for that. Okay, like I said, well, what I was saying was that it's the beginning of a new week. It's a Monday, and on Mondays, I like to start the week with a message of positivity. And this week in particular, and for the past few weeks, I think it's been important for me to acknowledge that there is a lot going on in the world between some of the uprisings that we're seeing across American cities, between the fact that we're in the middle of a pandemic still that has gotten less and less attention, and then also between Chadwick Boseman died last week, and so these things are impacting our mood collectively. And so, as your teacher, I just want to acknowledge that it's impacting me too. It makes me feel a little tired, a little hopeless at times, and so it's okay if you're feeling that way as well. But what helps me is being able to see things in a long-term vision, being able to recognize that every step along the way is not going to be easy. There will be days that are harder than others, but if I can remain rooted in myself and in my core values, and if I can remain surrounded by people who are going to support me towards that long-term vision, then ultimately I will get there someday. And I believe that. So I want you all to just take some time to think about whatever it is you're hoping to achieve this week. And don't only establish that vision for yourself, but start to think about some goals that you can set, and make them SMART goals, make them specific, make them timely, hold yourself accountable to meeting them. And after you've established that vision, but you've also established some strong goals for yourself, you'll be in a good place moving forward to actually achieve those goals. All right. So I know there's a lot going on. I know you all are young people who are balancing a lot. And so if ever there's a time you need to just speak with someone and you just want someone to just listen, just know that I'll be that person for you. But additionally, today and for the rest of the week, we're going to be wrapping up Unit 1 and jumping into Unit 2. Unit 2, I think, will be a bit easier for you all because you've been introduced to many of the concepts in Unit 2, either in middle school or last year during your first year of high school. But today we're thinking a little bit more about enzymes, and then we'll take some time to review some stuff that we talked about last week. The asynchronous portion of class, I think, is going to be most important today because you all should know by now that tomorrow, September 1st, is when all of your Unit 1 assignments will be due. So the asynchronous assignment today is relatively short, but the rest of that time should then be used to complete other assignments that perhaps you have not completed up to this point. So hopefully your weekend was restful. Hopefully it was rejuvenating, but hopefully it was also productive and you were able to get some things done. Excuse me. So the objective for today, biologists will be able to understand how to read an enzyme graph and determine an enzyme's optimal temperatures and pH. So graphing is not just for math. We do graph in science. In fact, science relies heavily on graphs and on hundreds of different types of graphs, because when you do scientific research and you collect data, you need to be able to represent that data in a visible way, not only for yourself, but also so that other people can better understand your findings and can agree with your conclusions. So that's why we graph in science. And many of the skills that you've learned about graphing in math can be applied directly to science as well. So can someone tell me which color in this graph, it may be hard to see, but which color in this graph represents the x-axis? Yes, thank you. Yellow. Yellow represents the x-axis here. We can also call the x-axis the horizontal axis. What about the y-axis in that case?
Purple.
Thank you. Purple represents the y-axis here, which we can call the vertical axis as well. All right, so keep that in mind. That's just useful to be able to orient yourself when you're seeing a graph. Excuse me. All right. So we saw a couple of these enzyme graphs last week, and we talked a little bit about them as well, but I wanted to reintroduce them here and talk about them further in greater depth. So please make sure that you have your notebook out and that you're able to take notes and that you're also able to refer back to notes that we took last week. I'll give you about 10 seconds to do that while I update my attendance because I see some folks have joined us. Okay. Excuse me. So we're seeing this red graph here. Now notice two things about the axes that we were just talking about. On our x-axis, we have pH. We talked about pH a little bit last week, but we see that it's increasing in intervals of 1 from 4 to 5 to 6, all the way up to 11. And then on the y-axis, we don't have any numbers, but we do know that the y-axis represents enzyme activity. So as I go up on the y-axis, as I go up vertically, what I'm saying is that my enzyme is becoming more active. It's working better, it's working faster. All right, so that means that as my red graph goes up, my enzyme is working better, it's working faster. And when it gets to the top, the very highest point of that graph, when it peaks, that's when the enzyme is working the best, at the very top of the graph. So if I want to know what the optimal environment is, first I need to find where is the enzyme working the best. So it's at the top of the graph, and then what I can do is just drop it down to figure out what the optimum pH is. And it shows me that in this case, the optimum pH is 8. Okay, so the enzyme is working the best when the graph is the highest. And when the graph is the lowest, aka when it's at 0, that means that the enzyme is no longer working at all. And we call that being denatured. So can someone tell me at what points does this enzyme denature? Where is this enzyme denaturing?
Like in the middle of 10 and 11.
Okay, so anything greater than 10 and a half, yeah, thank you. I think that was DeMarco. What about on this side? It doesn't have to be a precise
To be
number.
honest, it doesn't look like it denatures on that side.
It doesn't look like it denatures on this side. Okay, but we can see that it is hitting 0, right? We see that it's intercepting with the x-axis. So it's hitting 0. So that means that it is denaturing, and it's happening at let's just call it 4.75. All right. So anything below a pH of 4.75 is not going to work for this enzyme. That means it's just too acidic for that enzyme to work. Anything above a pH of 10.5, like DeMarco said, is also not going to work. That's just too basic. Okay, so the enzyme, this specific enzyme, whatever it is, it prefers to work between about 5 and 10. But it works best at a pH of 8, which is when the graph is the highest. Okay, let's try another one. We just answered that. So for this enzyme, we got a different enzyme, and we're looking at temperature this time. We're not looking at pH. But what would be the optimum temperature for this enzyme? Hello? What's the optimum temperature? This should be pretty obvious to you. 40 degrees Celsius. Thank you. So at 40 degrees Celsius, the graph is at its highest point. So all I need to do is just figure out where on my x-axis does that correspond to. So here it's very clear that they even gave us this dotted line. It's 40 degrees Celsius. Where does this enzyme denature, though? Where is it no longer working?
It stops working at like 50 to 60.
Good. So just above 60. Thank you, Brian. So let's call it 62 or 63. At that point, it's just too hot for the enzyme. It can't work at temperatures that high. So that's the point at which we would say it denatures. Now, we also should notice that in this graph, we had 2 points of denaturation. There's one over here and there's one over here. In this case, There's only 1 point because it only hits 0 in one place. So this enzyme, even though it doesn't work as well at lower temperatures, it can still work even if the temperature is 0 degrees Celsius. Okay. All right, so we talked a little bit about what you see on this slide and on Friday. But the question says, this diagram shows an enzyme substrate complex. Which of the following is represented by structure X? So here we have structure X. They're asking us, is structure X the substrate, the product, the enzyme, or the complex? I will say this is a very important question, and it is a question that will appear on. We will have a quiz tomorrow as well, specifically on enzymes. This question will appear on that quiz, and I can almost guarantee you that it will appear on your EOC exam at the end of the year as well. So at the end of the semester. So we want to be able to break this question down. Does anybody have any thoughts before I start or continue talking about what structure X is? Any thoughts? Okay. All right. So what we should remember about enzymes is that they are reusable, which means that if I use an enzyme in a chemical reaction, I'll be able to take that enzyme out of the chemical reaction after it's done, and the enzyme is unchanged. It still works. It's still the same shape, which then means I can use that same exact enzyme in another chemical reaction. And when that one is done, I can take the enzyme out again, and then I can use it in another chemical reaction, and so on and so forth. So enzymes are reusable because they do not change in the course of a reaction. So based on what I just said, enzymes do not change in the course of a reaction. Which of our structures would be the enzyme? Which one is not changing?
Wait, can you ask that again?
So the enzyme is the structure that is not changing in the course of the reaction. Which structure would that be in this diagram? Which structure is not changing?
Step 2 one.
What's that?
Step 2 one doesn't change.
Okay, so I'm looking for one of the structures. We've got 3 structures here: structure X, structure Y, and structure Z. Which one is not changing in the course of the reaction? From step 1 to step 2 to step 3. Okay, maybe I'm confusing you here. Structure Y, which we can see right here, is starting off in step 1 in the exact same way that it ends up in step 3. It's not changing. We see that we can see structure Y in step 1. We can see structure Y in step 2, and we can see structure Y in step 3, and nothing has changed about it. It's the same shape. So that means that structure Y is your enzyme. So we could cross enzyme out. Enzyme is not the answer here because they're asking about structure X. They're not asking about structure Y. Okay. So structure X appears in step 1, and then we see it again in step 2, but it's now attached to the enzyme. And then we don't see it over here in step 3. We don't see structure X anymore. So structure X is undergoing a change. Something's happening to it. But because it appears in step 1, that means that it is what we are starting off with. And the thing that we always start off with is called the substrate. Okay, so structure X is your substrate. It is your starting material. It's your reactant. So I'm going to type that into the, Alyssa, well, you could have saved me 4 minutes had you just taken yourself off of mute. But I see your answer now in the chat. Thank you. That's correct. So substrate is a synonym of reactant. Okay, so both substrates and reactants are the starting material in a chemical reaction. The opposite of that would be a product. Products are your final materials, chemical reaction. Okay, so in that case, structure Z, because structure Z is what we are finished with, or what we're left with when the reaction is over, structure Z is your product. The enzyme substrate complex, the complex is what we see in step 2 here. The complex is when, complex is the substance formed by substrate and the enzyme. So we can see that the substrate, which is structure X, just like Alyssa told us, and the enzyme, which is structure Y, have come together. They've created what is called the enzyme substrate complex. Okay. So if you are confused about that, I would definitely encourage you to go back and rewatch that Amoeba Sisters video from Friday. I would encourage you to go back and look at your notes from Friday. I would encourage you to watch the presentation from Friday or from today, just so you can really think about what the enzymes and how the enzymes relate to the substrates and the products. So this enzyme is digesting this substrate. It's breaking it down. We broke it down from one piece into 2 separate pieces. All right. So what do you all remember about nucleic acids? What is the monomer of a nucleic acid? What's it called?
It's called nucleotides.
Yes, excellent. Thank you, DeMarco. You're going to type it into the chat. Nucleotides. Spelling it. Nucleotides are the monomers of nucleic acids. And so this is a nucleotide right here, this entire structure. A nucleotide is made up of a phosphate group, a 5-carbon sugar, and a nitrogenous base. What do nucleic acids do for us? What's the function of a nucleic acid?
The function is it controls production of proteins and all cellular activities.
Good, excellent. So what I can tell DeMarco is doing is he's looking back at his notes from previous days, and that is exactly what he should be doing, and that's hopefully what everyone else is doing as well. So I want to definitely commend him for doing that and for taking good notes. So the function of a nucleic acid is to control the production of proteins. They are the instruction manual to make proteins. And we can also think of them as the boss. They tell the rest of the cell what to do. And can you tell me the examples, DeMarco?
Yeah, the examples, DNA and RNA.
Excellent. So we've got 2 examples of nucleic acids, and they are DNA and RNA. You should remember from one of the activities that we did last week, and hopefully you also have it in your notes, that there are some clear differences between DNA and RNA. Even though they are both nucleic acids, they do have differences. I'm not going to talk about those today, but hopefully you have them in your notes. So I'm not going to spend too much time here because in Unit 5, we will go into much greater depth about this, but DNA is found in a variety of different forms. And the two forms that we focus on in this biology course are chromatin and chromosomes. You do not need to write this down right now. This is just going to help set us up for the big point that you will write down. You can write this down, the parts that are highlighted. I would encourage you to write down the words that are highlighted in yellow here. But of course, DNA and proteins are connected. Like I said, and like DeMarco said, nucleic acids, like DNA, control the production of proteins. And they do this because we have billions and billions of nucleotides that make up our DNA. So a lot. But only a small portion of them would be responsible for producing a single protein. Like I said, the protein that gives us color in our skin is called melanin. And only a small portion of those 3 billion A's and T's and C's and G's that make up our DNA is actually required for producing melanin. Only a small portion of it. And that small portion is called a gene. So each gene is responsible for making a specific protein or set of proteins. Oftentimes, related genes will be next to each other. For example, the genes that are required to give your hair a specific color would be located next to the genes that are required to give your hair a specific texture. The genes that are required to give your skin a specific color are located close to the genes that are required to make your eyes a specific color. So related genes are located near each other along this long path of DNA that is 3 billion nucleotides long. So genes code for proteins. Now, there are large portions of our genes, of our DNA that we don't fully understand yet. We don't know exactly what they do. And as far as we can tell, they don't do anything. So actually, about 20 years ago, at the beginning of the 21st century, there was a project called the Human Genome Sequencing Project that literally mapped out the human genome. They mapped out the human DNA, and they were able to say, okay, this gene over here is responsible for height. This gene over here is responsible for, makes a woman more likely to develop brain cancer or breast cancer. This gene over here is responsible for detached or attached earlobes, and so on and so forth. All right, so they went out and they mapped the entire DNA. This was a very, very large project. It was extremely expensive. It required scientists from around the globe working together, but they've done it. And what they found was that big portions of of our DNA, even after they ran all these tests to figure out what they were responsible for, simply don't have any function, at least no obvious function to us. We can't tell what they do for us. And so this brings up a number of different questions, but primarily what this highlights is that our DNA is very much so connected to the DNA of other living organisms. And we're going to see that in a video that we'll watch after this. But here's the big takeaway, and this is definitely the part that you need to write down. Here's the big idea, and we're going to dedicate an entire unit to this idea, but it's good to write it down now. We start off with nucleotides, which, like DeMarco told us, are the monomers of nucleic acids. So if I take one nucleotide and I combine it to several other nucleotides, I can create DNA. That DNA then undergoes a process of conversion that we specifically call transcription. So DNA is converted into RNA, which is our other type of nucleic acid. And so in this RNA now are the instructions to make proteins. So essentially what we just did is, let's say you were interested in learning how to, you guys like YouTube a lot, so you basically can watch, you can find a video for anything on YouTube. But let's say you wanted to learn how to make a bed. And you watched it on YouTube, you watched a video of somebody explain how to make a bed. But before you actually wanted to jump into the project, you decided to write down each specific step and maybe even include some pictures in your steps. So you don't have to keep pausing the video, and maybe if your Wi-Fi goes out one day, you can still work on your project, you can still build the bed, and you don't have to go back to the video. So now you've written down all the instructions. So the original video is like the DNA. Then once you write down the instructions that you hear in the video, now you've got the RNA. In order to build the bed, which we could say is the protein, that final product, in order to build the bed, you need all of the materials. You need screws, and you need posts, and you need potentially some nails. So in order to build that bed, you need all those materials, and those materials are the amino acids. So the original video with the instructions is called the DNA. Once you write down those instructions, now you have the RNA. Once you use those instructions to gather your materials and start to put them together, those are your amino acids. And once it's finally finished and all of the materials have been put together, you formed a bed, which is your protein. All right, so that is the big idea here. That's the big idea. And you all can think about an analogy that works for you. That's really a key study skill, is if you can start to get creative and connect different ideas. All right, not doing that. Let's watch this video. We're perfectly on time.
In an organism's DNA is called its genome.
Sorry, I wasn't sharing my, I'll go back because I wasn't sharing my screen. I apologize.
DNA is the instruction manual for how to build life. From microbes to plants to human beings, it defines us all. The complete set of instructions encoded in an organism's DNA is called its genome and is passed from parent to offspring during reproduction. Information is stored in DNA using just 4 types of molecule which occur in pairs. There are billions of these pairs, organized in a double helix structure that is both strong and compact. These pairs also allow each strand to act as a backup for the other, a remarkably efficient way to safeguard this precious genetic information. DNA folds into paired packages called chromosomes that are stored in the nucleus of the cell. Different species have different numbers of chromosomes. Humans have 23 pairs. Chromosomes contain many genes. A gene is a section of DNA that holds the instructions for a protein. Proteins are essential for life and perform a huge variety of jobs, from controlling the function of a single cell to determining the shape of a whole organism. Within a species, each organism has very similar DNA. In human beings, the difference between one person and another is a fraction of 1%. But it's what makes us individuals, giving us different facial features, hair color, and height. The uniqueness of our DNA can be used like a fingerprint to identify us with an extraordinary degree of accuracy. By reading DNA, scientists have discovered that we share sequences not just with our own species, but with every other living thing on Earth. Chimpanzees, one of our closest living relatives, share about 96% of our DNA. But we also have genes in common with fish, plants, and bacteria. Powerful evidence that all life came from a single universal ancestor billions of years ago. We haven't just learned to read the instruction manual for life, we can rewrite it as well. People have been manipulating DNA since before we knew it existed, selectively breeding plants and animals to bring out desirable traits. Now, genetic engineering allows us to directly alter DNA in a lab, creating new varieties of life. From plants that can resist disease or drought, to bacteria that can mass-produce life-saving hormones. But we don't yet know what all of DNA does. Lengthy sequences make no proteins at all, and have perhaps mistakenly been labeled junk. Some people are worried about these gaps in our knowledge and unforeseen problems they believe genetically modified organisms may cause. What's clear is that the instruction manual for life is more subtle, elegant, and complex than we could have possibly imagined. DNA has revealed many of its secrets, but we still have much to learn.
Okay. Any questions or observations from that video? I think that's pretty cool stuff. I could also just be a nerd, so. Anything anybody wants to point out? Yes, no. Can I get some responses in the chat so I at least know you all are there? Thank you, Fanique. Thank you, Brian. Thank you, Makyron. Thank you, Joseph. All right, it's a little better. I know it's a slow Monday. Thanks, Sean. But we got to be somewhat engaged here. All right, let me go back to my slides. Okay, so that's it, and we're on time. I'm actually 2 minutes behind. So you all have 33 minutes left in this block to do your asynchronous work. So go ahead and knock out the exit ticket, and then also do, it's called enzyme practice on Canvas. And once you do that, I would definitely encourage you to try to knock out any work that you have not done up until this point. Remember, tomorrow is the last day of our unit. Thank you, Jaden. Thank you, Kesley. Tomorrow's the last day of our unit, so please make sure that you are on top of your work and are not trying to turn things in late.
I got a question.
Yes, sir.
When is the end of the quarter?
The end of the quarter is October, I want to say 15th or something like that. I can give you an exact answer shortly.
I got a question.
Yes.
So after unit 1, we can't turn in that work or something?
You can turn it in after unit 1, but it might be penalized depending on what the circumstance is.
What, the highest grade probably be like a 79 or something?
Yes.
All right.
The last day of the quarter is October 16th. All right. So I'll be right here. Jaden, you got a question?
No.
Okay. I'll be here. I'm just going to put myself on mute. Let me know if you have any questions about how to access anything. Again, you should be looking for the enzyme practice on Canvas.
My fault, Mr. Ritter kicked me out.
I feel smart. Ooh, I got an 86. What? Ooh, what y'all get?
Tell me the answers, bro. Tell me the answers, bro.
Bro, you gotta think, bro. Use your brain, bro. Bro, where'd he just go, bro? He disappeared.
folks, it's 1:20, so you're good to go. Please make sure that you are keeping up with your work and trying to remain as organized as possible. We do have deadlines tomorrow, so if you need help between now and then, hopefully you are in the Remind chat and you can reach out to me with any questions that you may have, or email or text message, whatever you prefer. But other than that, I will see you all tomorrow.